Hey, my name is Fredrik Schjesvold. I'm head of Oslo Myeloma Center in Oslo, Norway. We're presenting a poster on a very novel drug. It's the first time it's been presented. It's not only novel in that way, it's also a very novel principle. So this is a trispecific. Probably all in myeloma have heard about what the bispecifics have done. They are approved now, both in US and Europe and many other countries. Where you have one protein, antibody-based, that is targeting both the myeloma cell or the lymphoma cell and a CD3 T cell, immune cell. So what happens when you bind these together is that the T cell is activated, the immune cell is activated, and it kills the myeloma cell or the lymphoma cell. This trial was an attempt to increase even that efficacy. We know that there are very efficacious drugs, but they don't cure patients in late line still. So here they try to build even more potent T cell stimulator. So instead of having just one target protein on the T cells, they had two. CD3 that is commonly used, and CD28, which gives another signal for the T cell to be stimulated. And on the other side, you're targeting the myeloma and lymphoma cells. So the intention here was to have an even stronger T cell stimulation, activation, so it kills more myeloma cells. For myeloma cells, they were targeted by, and lymphoma cells were targeted in CD38. It's the same target we use for daratumumab and isatuximab and these antibodies. And it's also present on lymphoma cells. So there was a valid sort of possibility that this increased T cell activation would kill more of the cells. So the study is complete. It's a phase one study, a dose escalation study. There were most myeloma patients, 40 myeloma patients and 7 lymphoma patients, not Non-Hodgkin's lymphoma. So there are so few, so it's difficult to discuss them, so I'll stay with the myeloma patients. But the surprise, the results were not as we expected, actually. So what happened was that the T cells were they were very heavily stimulated, but so much that they got exhausted quite early. So what happened was that we do see in bispecific treatments, all the T cell treatments, we do see some CMV reactivations. It's difficult to clear a rhinovirus or a COVID virus, but still it's sort of manageable. It's mostly blood samples and light symptoms. Here we saw real escalations of even CMV, but also EBV, Epstein-Barr virus, which you don't see so much with the other bispecifics. So it seems like this double targeting of the T cells is just too much. The T cells just activate and then they just exhaust. And I think that also is probably explanation that the response rates were quite low. I have to say that all patients were CD38 treated from before with daratumumab or isatuximab, and CD38 was the target here. So that's also a possibility. So the efficacy and toxicity are two different questions, sort of. But the toxicity with this exhaustion and virus problems turned out to be so huge that the drug is not developing further. This presentation here and the following publication will be the sort of end of this story. But I think it's learned us a lot about how also not to stimulate T cells, because stimulating T cells has been what we've been discussing for many years, and how to increase that. And this was not the way. Important to understand, because there was belief in this before we tried. In the lab it works, so that's often how it is. But here at ASH we will see a different concept using a trispecific. This was a trispecific with 2 heads on T cell stimulating extra much and 1 on the myeloma cell. Here we will see the opposite, where you have 1 on the T cell but 2 on the myeloma cell. And that seems to be a better way of doing that. So that trispecific coming towards CD38 and BCMA on the same cells, in addition to stimulating T cells as usual. So we have learned now this is more probably the way to move forward with that. If you have a B cell, not a good B cell function, you get bacterial infections. You get pneumonias and streptococcal diseases. So very common in myeloma. So we substitute the bispecific patients because they're so efficient against plasma cells that they kill all the antibody production. So we use immunoglobulins for that, but for the T cell specificity, that doesn't help. The antibodies doesn't help against intracellular agents like viruses. Then you need T cells to work. And if you have a treatment that blocks out both T cells and B cells, then it becomes difficult for the patients. I I think there's several things to understand about the study. One thing is that when you run out of options, phase 1 trials that is left when we don't have any more standard treatments. Phase 1 studies have the great advantage of offering you a benefit when there's no other possibility. So I think patients should know that, and also know that most phase 1 trials fail. But in my opinion, it's always better to try this, because you can also be the patients that gets the first possibility to use the drugs that we now use in the clinic before they get there. So to participate in phase 1 studies is a really good thing, but it's okay to also know that they are the least secure thing in treatment. We don't know before we start, and everybody has to just accept that. But also phase 1 study patients are being even more taken care of than other patients, because we know so little, and very few centers do phase 1 studies, selected centers.